Hi, my name is Elaina Greaves, and for my master's thesis, I'm looking at the impacts of hybridization on native crabapple Malus coronaria by domesticapple Malus domestica in southern Ontario. So introduced species are species who are established outside their natural range, either for agricultural purposes or feral, and can have impacts on the native biodiversity. So one such impact is hybridization, which is the mating of closely related species to produce an offspring with some level of viability and fertility. Hybridization is important for evolution, however, impacts of this process are poorly understood. From a conservation perspective, there are three main points in regards to hybridization on rare native species. The first one is demographic swamping, also known as seed discounting in the literature, is when a non-native pollen fertilizes ovules that would have originally been producing conspecific seeds, but now are producing hybrid seeds. Pollen limitation is a factor that works with demographic swamping and is when a flower does not receive enough pollen to fertilize all the ovules, so the flower produces less seeds. In other words, the amount of pollen determines the amount of seeds a flower can produce. The second mode of hybridization is genetic assimilation, also known as genetic swamping or genetic introgression in the literature, is when a small native population mates with a big non-native population to create hybrid individuals who now have the ability to mate back with the native and non-native populations, until eventually there's only non-native populations and hybrid individuals left, with some sort of genetic code of the native species. There is very little information in the literature of the impacts of these two processes on native species survival. Hybridization between species can also be associated with asexuality, and these associations can be strong, but most are studied in a lab setting instead of a natural setting. However, the mechanisms underlying the greater occurrence of asexuality in hybrids is not fully understood. This association does create a possibility that asexual seed production rates can be influenced by hybridization. So my research focuses on the impacts of hybridization between the native crabapple, Malus coronaria, which is the picture on your right, and the introduced domestic apple, Malus domestica, which is the picture on your left. Domestic apples are a cultivated species and is one of the most important and widespread fruit crop in the world. It can exist either in commercial form or as feral trees out in the wild. It is a diploid, self-incompatible, and insect pollinated. Crab apples are native to southern Canada and are relatively uncommon. They are tetraploid, self-compatible, and insect pollinated, and can reproduce sexually and asexually. What is very interesting is that in the field, crab apples are known to be genetically uniform within populations, even though it can produce many different ploidy offspring. And ploidy is a set of chromosomes in a cell. The ranges of these species overlap, as well as flowering times and pollinators. The gray part in this map is the range of crabapple species. The squares represent the domestic apple range, and the squares with dots are orchards of domestic apple species. So this figure was used in Cron and Husband 2009 paper. Malus species also have the ability to reproduce sexually and asexually. This is an example of sexual and asexual, or what is also known as apomixis reproduction, with a domestic apple pollen grain and a crabapple flower. So as you can see on the left-hand side, it's sexual reproduction. So we have the domestic apple pollen grain, and each of those 1X's with the arrow represents a sperm nuclei. And each of these sperm nuclei, one fertilizes the embryo, the other fertilizes the endosperm. So the offspring would then equal a 3X embryo and a 5X endosperm. On the other side, which is asexual reproduction, same thing as before, we have a domestic apple pollen grain and 2 sperm nuclei, but instead of one fertilizing the embryo, they both are fertilizing the endosperm. So then the offspring would be a 2X embryo and a 6X endosperm. In addition, there's a possibility that asexual reproduction rates can be influenced by hybridization of different ploidy species. So now I'm going to talk about my research questions and hypotheses. Overall, I want to examine the impacts of hybridization by domestic apple on the native and uncommon crabapple. So my first question is, does hybridization by domestic apple pollen negatively influence the number of conspecific or same species seeds produced by crabapple? So this is looking specifically at demographic swamping and pollen limitation. So my hypothesis is the effects of domestic apple pollen on conspecific seed production in crabapples will be determined by the strength of pollen limitation. And then my second question is, what is the contribution of hybridization to a high degree of genetic uniformity in populations of crabapples? So this is specifically looking at asexuality. And my hypothesis is hybridization enhances genetic uniformity caused by a disproportionately high number of asexual embryos in crabapples. So I'm just going to go over my methods. So my study site was at the Rare Charitable Research Reserve in Cambridge, and on this site I pollinated 26 crabapple trees. I did not do any domestic apple trees. This is strictly, I'm doing research on the crabapple trees. And here is just a general idea of where these trees were located. We had some fun naming all of the trees, so yeah. So for my fieldwork, 3 branches were tagged on at least 3 sections of each of those 26 trees, and there were 4 treatments done on each of the trees. So 1 branch in each section had an open pollinated treatment, which the flowers were tagged but then left alone. 1 branch had a supplemental crabapple pollen treatment. So these flowers had crabapple pollen added to them but then left alone. And the last branch had the last 2 treatments. So 1 was just adding crabapple pollen and 1 was just adding domestic apple pollen. So this branch was bagged like you see on the picture below, and the flowers were emasculated, so I was removing the anthers and then pollinated with either crabapple pollen or domestic apple pollen. At least 3 flowers were chosen per treatment on the branches, and mature fruit was then collected at the end of August. So back in the lab, the fruit was cut up and then the seeds were removed and counted. The second step of that was then we did Flow Cytometric Seed Screening to determine the DNA content of both the embryo and endosperm of all seeds, and then the ploidy was determined from that. So with that in mind, here's a table of the possible ploidies that an offspring can have. On the left-hand side with the egg, that is strictly a crabapple egg, and the 1N or 2X represents a reduced egg, so meiosis was completed, and then the 2N, 4X is an unreduced egg, so meiosis was not completed. The second column with crabapple pollen, this is when crabapple pollen is added to the seed, and these are the possible combinations. The small circle is the embryo and the bigger circle is the endosperm. And then it's the same for the next column, which is strictly domestic apple pollen being added. And then it's split into sexual and asexual seeds. So I'm just going to go through a couple of my results that I have so far. So before I continue with those results, I want to clarify something that will come up during this talk. When I'm talking about the seed, it is in reference to both the embryo ploidy and the endosperm ploidy. So specifically, what type of pollen fertilized the seed. So that is the diagram on the left. When I'm talking about the embryo, I will be referring to how it will end up as a seedling. So just the ploidy of the embryo, with it, which is the graph on your right. So with that in mind, all of the asexual seeds, when domestic apple pollen is present, will be considered as conspecific embryos. Yeah. So these 2 graphs are only using the open and supplemental pollen treatments. So the first 2 treatments, this is what the results say. So with looking at specifically the conspecific and hybrid seeds, so this is both the embryo and the endosperm, we can see here that there's about 27% are actually hybrid seeds, which I find kind of cool. And then looking over on the graph to your right, you can see that out of that 27% hybrid seeds, only 14% are actually have hybrid embryos, which I find also kind of interesting. And then this data is from all of the seeds. We're just looking at sexual versus asexual seeds. And as you can see here, about 49% of all of the seeds are asexual, which I find pretty interesting. And so then looking specifically at my questions, question 1 is relating to demographic swamping and pollen limitation. So this data will only be on the open and supplemental pollinated treatments. And when looking at the seeds, so this is with the embryo and endosperm ploidies, as you can see, there's really no significant difference between the number of conspecific seeds and the number of hybrid seeds in a specific fruit. And then when we're looking at the embryo specifically, so just looking at the embryo, as you can see, there's also no significant difference between the two. And then looking specifically at pollen limitation, there's no significance regarding pollen limitation with crabapples. And why there's no significance, it could be due to crabapples having a low viability in pollen, but this will have to take more investigation to come to this conclusion. So then looking specifically at question 2, which is asexuality, this data was completed with all of the treatments, so the crabapple, domesticapple, pollinated treatments, and then open and supplemental pollinated treatments. And just showing you the graph on the left is just looking at the number of seeds from asexual and sexual with crabapple pollen and domesticapple pollen. And as you can see here, there is actually a significant difference between asexual and sexual seeds. However, it's in the opposite direction that we thought. With this information, we can say that hybridization doesn't actually affect the asexuality of the seeds, which is kind of cool. So in conclusion, about 27% of the seeds were hybrid seeds. So remember, this is the embryo and endosperm ploidies. And only 14% of the seeds actually contained hybrid embryos. In regards to question 1, hybridization had no impact on conspecific seed production, so demographic costs are negligible. And then in regards to question 2, hybridization is associated with the shift in seed origin. However, in contrast to our hypothesis, asexual seeds are less frequent with hybridization. So I'd like to say a few thank yous to the helpers in the field who have helped me pollinate the trees and also find the trees. And then to my supervisors, Brian Husband and Paul Cron, who have helped me kind of throughout this entire process. And here are a couple of funding resources that have also helped me being able to do this master's thesis. So thank you so much for listening, and I hope you have a great day.